Hi, welcome to my biology class. The previous video we have seen the outline classification of Pittacus, 5 kingdom classification, and from today on actually we will be elaborating one kingdom after other. So we start with kingdom Monera. See, Monera means all prokaryotic cell. All prokaryotic cell comes under Monera, and this you have to go through bacteria, cyanobacteria and mycoplasma. And bacteria, when you talk about a bacteria, it is such a small creature and the average size is 2 μm only. Average size is just 2 μm. But you see, they have got an extensive metabolism. Extensive metabolism and very complex behavior. Now, the extensive metabolism means, see, metabolism, the major metabolic activity of any living cell is production of ATP. So, here the ATP production can be carried out either by the way of aerobic condition or anaerobic condition, or they just go with photosynthetic, chemosynthetic, and they are heterotroph and they are saprophyte, they are symbiont. So, they are capable of living in any kind of condition and they make ATP. That is why they have an extensive metabolism, such a small cell, but they are adapting themselves to any kind of atmosphere to make ATP. In that, you have two major groups. One is Archaebacteria, another one is Eubacteria. Archaebacteria, they are believed to be the first living creatures in this planet Earth. So they've been living in so long time. So they had undergone a lot of changes in their life journey. So they were able to fit themselves in any kind of natural calamities. So accordingly they fit, and so they are capable of living in any extreme condition. Any extreme conditions. So when you talk about extremophiles, that is Archaebacteria, they are acidophiles. They will be living in a complete acidic environment. They are surviving. And thermophiles, extreme temperature, 100 degrees C, they will be able to live. And alkaliphiles, alkali nature, the soap solution you are just throwing on the soil, and they are able to live in that area. And methanogens, methanogenic bacteria are strictly anaerobic and they are able to live in a ruminant animals, like the gut chewing animals. They release methane gas, biogas. And psychrophiles, psychrophiles in the sense they live in a 0 degree centigrade. So extreme cold condition, Himalayan ranges, Alps, snow covered mountains, and these are the ones. So they are in extreme weather condition, they are able to fit themselves to live, and so they are generally called as extremophiles. The major difference between Archaebacteria and Eubacteria is, eubacterial cell wall is peptidoglycan. It is not cellulose, it is peptidoglycan. But Archaebacterial cell wall is not made up of peptidoglycan. It is made up of a cellulose and some protein are present in it. That is the major thing. And when you talk about a protein synthesis in future, I will explain more of a human protein synthesis correlate with Archaebac rather with Eubacteria. Okay. When you talk about a eubacterial cell, the true bacteria, generally the classification goes like this. Bacterial cell structure and other things, the complexity, everything we'll see under the cell topic. And here the bacteria can be classified on the basis of shape, basis of flagella, stain nature, respiration mode and nutrition mode. And here shape, cocci, it is spherical. Bacilli, rod shape. Spirally, spiral shape. Vibrio, comma shape. Okay. So these are the shape. Generally any bacteria belong to this category, either they are spherical or rod shape or vibrio kind. When you talk about a flagella, sometimes the bacteria without flagella, there is no flagella. Flagella meant for their locomotion. So sometimes there is no flagella at all. A trichus. Trichum means hair-like growth. A means absence of trichum, A trichus, no flagella. Monotrichus, only one flagella in one point, one particular point in their body. And Lopotrichus, a tuft of flagella on both the sides, And Amphitrichus, one flagella on either side. Amphi means two, one flagella on opposite direction. And Peritrichus, flagella present throughout, it is present throughout the body. And Cephalotrichus, a tuft of flagella in one side, okay, this is more or less like this. And when you talk about the strain, say when you are working on bacteria, first and foremost what you have to see is whether it is gram positive or gram negative. Okay, so Graham's strain, it is Dr. Scientist Graham's name. So he, see the bacteria which is seen in violet color or purple color after the alkali wash, after 2 stains, and that will be a gram positive. If the stain goes off and it will be looking like a red color, and that is gram negative bacteria. So with this you can identify. So generally they belong to positive and negative. Gram stain is the major thing when you are working on to classify the bacteria. And when you go with the respiration, it goes like aerobic, anaerobic. With the help of oxygen they respire, without oxygen they will respire. Most of the bacteria are working for dead and debris, and they decay the content. They all go with anaerobic kind. And when you talk about the nutrition is variety. They are autotrophic bacteria and heterotroph. When you talk about an autotroph, they are photosynthetic. They have a bacterial chlorophyll and they carry on their life. And they have chemosynthetic bacteria. They use nitrogen and other things, and they prepare their own food. And heterotroph means they don't prepare the food, but they take from other sources. In that they will be a parasite or saprophyte. You know what parasite means? Parasite is they are living on the living cell, and they engulf the content present in the host body. And saprophyte means they depend on the dead bodies. So they are able to live in this. And apart from all this, there are bacteria as a symbiont, like they are symbiotically associated with other living creatures. So they are able to carry on their nutrition in different modes, so that they will be able to prepare ATP. So with this the general account of bacterial cell. Bacterial cell details, okay, what is the size and cell wall, capsule, and the content, the vacuoles, whether it is a sap vacuole or gas vacuole, nucleoid, everything will deal under the cell title, prokaryotic cell and eukaryotic cell. From here we will be moving on to cyanobacteria in next video. Okay, so with this I'll put a full stop, and if you have any comment, you please pass it.